This is the part 3 of the microscopy, wherein we are going to discuss about the measuring of the cell. Here we are going to discuss about the unit conversion, how to measure cell from a photomicrograph using a ruler, how to calculate magnification from the scale bar, and how to calculate or measure the cell using a microscope, that is using an eyepiece, graticule and stage micrometer, and how calibration goes hand in hand with them. So first and foremost thing is unit conversion, which you should know. So like before going into converting the units, let me just revise you the concept of magnification. The formula of magnification is, it is I by A. Okay, I represent the image size and A represents the actual size. How I ask my students to remember it as IM, simple way. And then you can rearrange it for like how we have rearranged for magnification. Similar way you can rearrange for the actual size also. So IM is the simple way to remember the formula of magnification. Now remember, whenever you are, image size will be generally the one which you are measuring with the ruler. That will be mostly in millimeters, and the actual size will be much lower than that, which will be in the micrometers unit. So here the two units are different, so conversion of units is very important math required over here. So here is the simplest unit conversion chart, which you can remember it. Generally millimeters to micrometers are generally used. So whenever you are going from millimeters to micrometers, you multiply by 1000. And whenever you are going backward, that is from micrometers to millimeters, divide by 1000. You can just remember it in a sequence, and you can just remember the arrows. When it goes down, it goes multiply. When it goes up, it goes division. Okay, it's again the math over here. So rearranging and unit conversions are very important. Two units should be same, and generally they will ask you to find out in the micrometers. So whenever you are finding millimeters, just multiply it by the 1000. So let's start with the first scenario of measuring the cell. So here is, for example, magnification is provided to you 100. Image size is given to you, which is 50 millimeters. You need to find out the actual size. So again, IM, you can make a subject of A, because actual size is the what you are finding right here. So A goes like I by M. I is what? 50 millimeters, and M is what? 100. So that will be 0.5 millimeters. Here they have not asked you to convert unit into micrometers or anything, so just keep it as it is. For example, another one also I have for you guys. So here also they are asking you to calculate the actual size, and the picture is given right here. So first of all, what the first step is, measure the photograph which is given to you. Okay, micrographs rather I would say, because it is taken using a microscope. So micrograph which has been given to you, measure it with the ruler. Remember it's in mm, and that is 80 mm right here, and magnification is given over here, which is around 90,000. So again apply the formula. IM, A you want to find out, I and M are given. Image size in millimeters is 80 mm, divide by magnification. That will provide you 8.9 into 10 raised to minus 4 millimeters. Now what you are supposed to do is you are converting it into micrometers. So what you will do is you will just multiply it by 1000. That will provide you 0.89 micrometers. So whenever you are going from mm to micrometer, remember what you are supposed to do, multiply. when you are going back forth, division. Here we are going from mm to micrometer, so it is multiplication. So your answer is 0.89 micrometers. So I hope this first step is easy. Two things will be given, one thing you need to find out. Actual size over here was missing, so first step was finding the length using a ruler of this micrograph. Magnification is also provided. We have kept the formula, we have checked the units, we have converted the units in the micrometers, and here we are. Okay, and if you want the number in the whole numbers, okay, 0.89, then you can just convert it further on into nanometers, which is 890 nanometers. So this is how your first scenario goes on. The second one is using a scale bars. Scale bars will be given to you on the micrograph. Say for example, the scale bar given to you over here is somewhere around 6 micrometer. So this is the scale bar. This scale bar will be provided on the micrograph, so which is 6 micrometers. So first step, what you will do is you will take a ruler again and you will measure the length of this scale bar, like how you was measuring the length of the micrograph. So here what you are going to do is you are measuring the length of this scale bar. So when we do that, it will come around somewhere 3.6 centimeter, that is equal to 36 millimeters. So length of the scale bar is 36 mm. So what you will do next is convert millimeters into micrometer. First step, measuring. First scenario, we were measuring the micrographs. Second scenario, since the scale bar is given, we are measuring the scale bar using a ruler. First step is measurement done. It is in mm, converted into micrometer. So that will lead me to 36 into 1000, which is equal to 36,000 micrometer. Okay. Now, why we are converting it? Because here on the scale bar it is micrometer. Okay. So we have converted it. Now what you will do is put it in the formula. Magnification is I by A or IM. M subject, I by A. So what is the magnification? Image size will be the one which we have calculated using a ruler. And the actual size is given 6 micrometers. On the scale bar, this is the actual size, this is the image size. We need to find out the magnification. So units will get cancelled and you will get your answer, which is 6,000. Now remember why you need to write it down times, because you are calculating the magnification. So always write your answer like this, 6,000 times magnified. So this is the second method of calculating a magnification using a scale bar. Scale bar will be provided to you on the micrograph itself. So steps remains the same. First of all, when scale bar is not given, you will find out the micrograph size using a ruler. Here also same thing, you are finding out the size of this scale bar using a ruler. Then you are converting the unit and then you are applying the formula IM. Whatever is missing, just make the subject. And here you get your answer. This is the second one when scale bars are given on your micrographs. So see how they are given. I have another example for you guys. Yeah. See. Here you see the scale bar is given 50 micrometers, which is your A, actual size. Then the measurement with the ruler has been done. Okay, that is 20 mm. Conversion and everything you need to do it, and you need to put it in the formula and you will find out your magnification. So this is how the scale bar will be given on the micrograph. When scale bars are not given, you simply need to calculate the micrograph which has been given to you. So I hope you are clear with the second scenario also. Now for your practice, you can just print out this different scale bars, okay, and you can calculate magnification for this scale bars. Okay, these are the different scale bars given to you. So you can print out this and you can practice it for the scale bars, that how this works. So this is how you can also make easy way for the conversion of the units also, and you will learn it also. Now third most scenario is using a eyepiece, graticule and a stage micrometer. That is, you need to measure a cell using the graticule. So basically this is also helpful in AS level practicals where they will ask you to measure the cell using the microscope. So these are the one important practical skills also. So please you guys focus right here. So graticules are what? They are more suitable ruler for measuring the cell. Till now we were measuring it with using the ruler, okay, the normal one which we are using in our day-to-day life. But this graticule is the one wherein microscopes are used. So here two things are required: slide graticule and eyepiece graticules. Two different things will be there. So stage graticules are a precise measuring device with small scales that are placed on the microscope stage, and they are used to calibrate the value of the eyepiece division at different magnification. So slide graticule is generally your stage micrometer, which is used for calibration purpose. Why calibration is required, we will see in the next slide. But for now, what is stage graticule? It is a precise measuring device. Please remember this, okay? They have the precise measurement given on it. And the eyepiece graticules are the one which are fixed on the eyepiece lens of the microscope. Okay, you are familiar with the ocular or the eyepiece lens, which we have discussed in the part 2. So if you haven't checked that, please go and check that, because your eyepiece graticules will come under that. Okay, they are fixed on the eyepiece lenses. So they are what? They are a measuring device that is placed in the eyepiece of a microscope and act as a ruler when you view on the object under the microscope. So we have the eyepiece, right? So why we need a stage graticule? Why we need a stage micrometer? So let's discuss that why stage micrometers are used. So there is a need of the stage micrometer is because your eyepiece graticule, when you observe it, it looks like this. So you see there is arbitrary units. Okay, there is, you can call it as a no units given. So how you gonna measure the cell? So your eyepiece graticules, they have the arbitrary units on it. Okay, there are maybe no measurements, no units will be given to you, which is this part. So in order to know that what is the size of our cell which we are measuring, we need to calibrate it using a stage micrometer or a stage graticule. So your stage micrometer scale is generally marked in 0.01 millimeters and 0.1 mm divisions, which will be given to you, which will be provided to you. Okay, so they are generally in 0.01 millimeters, that is about 0.1 mm division. So now what you will do is you will just superimpose both of this. That's what a calibration is. You superimpose this 0 with this one. This is the eyepiece graticule scale and this is the stage micrometer scale. So when you just superimpose both of them, you will have some divisions right here. You will have the units, because on eyepiece there was no units, but now you can have something to compare it off, right? So that's what the calibration is. Calibration of an eyepiece graticule. It's required because eyepiece graticules scales is very arbitrary. The units are arbitrary. So to calibrate the eyepiece graticule, a miniature or a transparent ruler known as stage micrometer scale, it is placed on the microscope stage. and then it is brought into the focus. The divisions will be provided to you, so that no need to worry. So this is how your stage micrometer looks like, and you place it on the stage right here. Okay, so you see this round part, there is a scale given to you, so you superimpose it with the 2 units, and there you go, you have the calibrated eyepiece. Now you have some units to your cells or organelles which you are measuring it. Now I want you guys to note this point, that the scale of the eyepiece graticule is not fixed. Yeah, the eyepiece graticule which you have placed, it is fixed, but that scale is not fixed. Why? Because your objective lens has different magnification. So when you change the magnification, the calibration also changes, which you can observe it through the picture right over here. Okay, it is less magnified image, maybe it's in 10x, this is 40x. So in 40x, if you see, the scale has been changed. So now you need to calibrate it again. So whenever you are changing a magnification, you need to calibrate it each and every time. Okay, because the scale, that does not remain fixed. Since you have changed the field, the magnification is changed, again you need to calibrate it, because maybe over here you have superimposed your stage micrometer here, and now when you have changed it, maybe it has shifted from here to here, so it is not calibrated anymore. So you need to calibrate each and every time when you are changing the magnification. Do not forget this. Okay, I hope I am clear with this note. You need to make a note of it. Students make most mistakes over here. Okay, they change the magnification and then they keep the same calibration, which will lead you to the wrong measuring of the scale. So please make a note over here, pause this video, make a note, and there you go. So now to understand more about the calibration, let's discuss one of the work example. Okay, so let's see this calibration with the worked example. Okay, so this one right here, this one is the eyepiece graticule. So your eyepiece graticules, which is placed in the eyepiece of the microscope, which is observed like this. Okay, remember, they have the arbitrary unit. Okay, you cannot find out the actual measurement with this eyepiece only. So what happened is, the second one, the cheek cells on the slide, okay, on the stage of the microscopes. There is a slide on the stage where cheek cells has been prepared and they have been focused on. Okay, so eyepiece graticule in the eyepiece of the microscope is this, and this is how you will observe your cheek cells when you have kept it on the stage under a microscope on the slide. Now if you have noticed, one cheek cell is somewhere around 20 units, because each one is showing the 10 units. So 0 to 10, 10 to 20, 20 to 30, 30 to 40. So over here, what thing you have recorded is 40 to 60. That means 20 units. Yeah. But what's the actual unit? 20 units is not enough. Okay, 20 divisions, what that means? What is the actual size, actual scenario of this cell? We need to find out the precise measurement, right? So now what you will do is, you superimpose the stage micrometer. Okay, right here. See. So whenever you are seeing, see from the diagram itself, you have your 0 right here and 100 maybe somewhere here. So your 100 divisions of what? Eyepiece graticule. So I'm just writing EG, eyepiece graticule, is equal to what? 0.25 millimeters of your stage micrometer. Why? Because your stage micrometers are marked in 0.01 mm, or you can just call it as 0.1 mm division. Okay, one division is 0.1 mm. So this is 2 and this is half away. It's not full division, you see. So it is 0.25 right here. So your 100 division of eyepiece graticule is equal to 0.25 mm. Okay, so just make a note. First of all, you have seen it's 20 units, but there is no measurement, no units are there. We require the units since we are measuring the cell. So what you will do is you calibrate it, you superimpose both of the scales, and you see the 100 division of eyepiece graticule is equal to 0.25 mm. I hope you got this. Again, see, they have already given you, whenever this slide comes, the stage micrometer, they will provide you that how many divisions are there on this slide. So don't worry about it. Okay, so this is already been marked in 0.01 mm, or you can just call it as 0.1 mm divisions. So 0 to here it is 0.1, 0.2, and this is half only. 1, 2, 3, 1, 2, 3, 4, and 5. So it is 0.25. So your 100 eyepiece reticule is 0.25 millimeters. Of what? Of stage micrometer. Okay, now if 100 is equal to 0.25, then one eyepiece graticule means how much? So I hope you know the cross multiplication. One eyepiece graticule into 0.25 mm divided by 100 eyepiece graticule, which will give you somewhere around 0.0025 mm. Okay, now convert this into micrometers. You always do that. So after converting it, what you will do, you will just multiply it and you will get 2.5 micrometer. Yeah, so here you go, your one eyepiece graticule unit. Okay, I should measure over here, unit everywhere is equal to 2.5 micrometers. And here we have noticed the one cheek cell. Right here it is 20, 40 to 50, 50 to 60. So it is 20 eyepiece graticule unit will be how much? So that will be 20 into 2.5 divided by 1, which is somewhere around 50 micrometer. So your size of the cheek cell is 50 micrometers. Here you have the unit now. Yes, I hope I am getting clear. First most role, first see the eyepiece reticule, you are observing it. Second thing, just take a note of your lens, that is the cheek cells. Remember to specify the magnification. At one point, magnification is important, right? Because at each and every time magnification, if you are changing, again you need to change the calibration. So first of all, check out that your measurement of the cell, then calibrate it. Check out how many 100 divisions will be your stage micrometer division. Note it down that if 100 eyepiece reticule unit is over here 0.25, then one eyepiece reticule unit will be how much? Whenever you find it out, then convert the units into micrometers. Okay, then since you have your units right here for the cheek cell, which is 20 eyepiece graticule units. So if one eyepiece graticule unit is 2.5, 20 will be how much? So that will be cross multiplication. Okay, so 20 into 2.5 divided by 1, which will be 50 micrometers. So your size of the cheek cell is 50 micrometers. Okay, I hope I am clear with this whole scenario of calibration, why calibration is required, and once you have calibrated it, how you can measure the cell. You just need to compare. Okay, so I hope this whole video series part 1, part 2, part 3 has been helpful for you guys. If that's so, please like my videos and subscribe my channels, and you can also comment in the comment box wherever you want more. Okay, whichever the topic you want the video on, you guys can suggest me, I can make the video for the same. Okay, And for any queries or anything, you can comment it anytime you want. Write down in the comment box. So please keep on suggesting me for more videos on what topics you are finding difficulties in, and for any queries on any of this series video, you can comment it down. I will answer it to you.